Today, I want to tell you about the work we've done in the last decade or so in developing spatial technologies that allow us to look at genomic information directly in situ, in the tissue. And I want to talk about some of the work from this technology that we learned that allow us to extract new biological insights beyond just looking at tissue organization. So, why do we want to do tissue spatial biology? Well, I will argue almost every biological system is spatially complex, right? From brain to embryos to cancers. When you talk about hot versus cold tumors, it's about the microenvironment and the position of the immune cells in the tumor. And so we want to be able to interrogate inside the tissue directly and see what the gene expression patterns are and how the cells interact with each other. And in the last couple of years, there's been a huge explosion in spatial technologies. And I think of them roughly in three rough categories. One is using in situ sequencing. One is using array-based methods where you transfer the tissue material onto microarrays. And then I'll focus specifically on multiplex FISH approach, which uses in situ hybridization and scale that up to the transcriptome level. And I think the multiplex FISH approach is the most accurate and highest resolution. But of course, there are some drawbacks as well. So the technology itself is built upon single molecule FISH, which was pioneered by Rob Singer and Arjun Raj, using short oligo probes that then go in, hybridize in tissues, and give you these distinct spots in cells when you look at them on a microscope. And then to give you a single molecule counting of the transcripts directly in the individual cells. So how do we scale this up, right? Because typically in a microscope, you only have 3 or 4 different colors. How do you scale this up to tens of thousands of genes? So the method we came up with is called sequential FISH, where we use sequential rounds of hybridization so that in the cell, even though all the dots are shown up in a few different colors, you can do multiple rounds of hybridizations. And for example, this dot here switches from blue to red to yellow and then to green. So each gene gets a unique barcode across multiple rounds of hybridizations. And this way, you can have a barcode list. And if you start with 4 different colors, if you do 4 rounds of hybridizations, you can get a couple hundred different genes encoded. If you just do 8 rounds, you can cover now the entire transcriptome. Now, so we showed the proof of principle experiments in yeast just with a dozen genes. So we can see, on the first round of hybridizations, you can get these colors. Next round, you switch colors. To zoom in pictures here, you can see these yellow dots switch to blue. And then there's a third round is error correction that goes back to the first round and allows you to correct for mistakes in some of the drops. And then a couple years later, we showed that we can actually scale this up to the transcriptome level for nascent transcripts by targeting intron regions. And so this is an image where this is a single nuclei from a mouse embryonic stem cell. And each one of these colored dots is a single transcription active site in the cell. So there's over 10,000 of these genes profiled. The white is the nucleolus, and the blue are the heterochromatic regions. And you can see that the nascent transcription happens essentially uniformly across the entire nucleus. And these dots are color-coded according to the chromosome. So the chromosome form distinct territories in their active sites, but it's basically, there's no major transcription factories, but they're all kind of spread out in the nucleus. And in some sense, we kind of cheated in this experiment because, the number of transcription active sites per cell is fairly limited, right? So we don't run into this density issue. And that's a problem with using optical microscopy to multiplex large number of genes, but that each one of these dots occupy a couple hundred nanometers in space, and the cell is only a few microns in space. So you can't really jam as many dots as you like in a cell. So we actually started working on this in this field by using super resolution microscopy to shrink the size of the dots and then allow us to pack more of the dots in. But it turned out at the time, the super resolution methods that have been developed use these stochastic blinking methods that require a single dye molecule to switch. And that's very hard to detect single dye molecules in tissues. So it turns out the sequential round of hybridization, the SeqFISH method, gives us a way to perform super resolution microscopy. Right? Think about it this way. If you hybridize against a subset of the genes, right, you can get these distinct spatially sparse dots, and then you can do Gaussian fitting to shrink the size of the dots. And then through multiple rounds of hybridization, you can then essentially sample all of the transcripts, all the genes, and each time you can compress the size of the dots to generate this compressed super resolved image. Right? In some sense, this is what we call deterministic super resolution microscopy. You can think of this as the difference between Illumina sequencing chemistry versus the PacBio. One is, deterministic, one round at a time, but a lot of signal. Other one is stochastic, or a single molecule, single dye at a time. So this is the method that allow us, right, then to multiplex now at the levels of tens of thousands of genes. Instead of scaling up, 4 to the 8th, now we can do a larger number of these, what we call pseudocolors, and then just do 4 essentially barcoding rounds to get to 20,000 genes. Okay, so apply this method to looking at mature transcripts now, and we can look at this mouse brain slice, zoom into this region, look at different cells, zoom into this cell, which is a pyramidal neuron, and then you can zoom in further and directly decode the different transcripts present in that particular region in the tissue. And each one of this grid is 100 nanometers, so we can really resolve the subcellular localizations of these transcripts in situ. And because you can now extract the transcription profiles of these cells based on the images, you can cluster the cells, get these different cell type definitions, and across the mouse cortex, you can get the different cell types, the neurons, the glia cells, and so on, and you see these organization in the tissue. Now, I think this gets to the sort of the struggle for the heart of spatial transcriptomics, is that, we make these pretty pictures. How is that fundamentally different from something that's just art, right? Like this pointillistic painting. You get a different type of people, different organisms, different developmental stages, right? Generating these images both take a lot of technical skill, but what is it that we can do beyond just the artistic representation of the tissue? Can we get some actual biology, right? And I will argue that artistic paintings are in some sense just a way to represent reality, but to do science, we have to discover some new things, new biology that is beyond just making a nice picture. So I hope in the next 10 minutes or so, I can show you 2 examples that we can extract new insights based on the spatial data beyond this atlasing effort. So one of them is looking at this kidney injury and looking at cell interactions, and the other one is zooming into individual cells and look at nuclear structure organization. So for the kidney injury, this is a collaboration between my lab and Andy McMahon's lab at USC, and it's really pioneered by 2 postdocs, Michal in my lab and Luisa in Andy's lab. And so in this particular case, we looked at a mouse model of ischemia injury of kidney, so basically choke up the blood supply to the kidney and then release it. And you wait 28 days, and we imaged sections of this injured kidney in 3 mouse, and then compared it to the healthy control, another 3 samples. So overall, there's 200,000 cells, and imaged about 1,000 genes or so. So one of these slices, sections is shown here, and when we combine all these data sets, we can get this cluster of the cell type definitions, which correspond to the known cell types in the nephrons in the kidney. So there's about 1 million nephrons in the kidney. Basically, you have blood vessels that go into these glomeruli, which then filters the content. It goes to these proximal tubules, then it's a loop of Henle, and then this descending loop. And so the spatial data allow us to cluster these distinct cell types, and they correspond to exactly where you expect them to be in the tissue. So you have the cortical proximal tubule cell localizations, and then you have the loop of Henle cells, which are more in the medullary part or the inner part of the kidney. So we noticed, of course, that there's an injured population that's present in the injured samples. It's not present, a little bit present in the wild type, but mostly, 90% of the cells are in the injured samples. And so how do we go, beyond this? So one of the questions is we can ask, what are the cells around the injured cells, right? So to do that, we can define a neighborhood. So we have a cell by gene matrix based on the spatial transcriptomics, but now we can convert that into a cell by neighbor matrix, where you say, okay, around these cells, what is the composition of the neighboring cells around it? And based on that, you can recluster the cells and arrive at these microenvironments or neighborhoods, and transform this tissue map from the cell type map to a neighborhood map. Okay, so what does this tell you? Well, what you can do is you can say, okay, now I break the tissue into these distinct neighborhoods. I can see that many of the neighborhoods comprise of, one major cell type, but then also most of these neighborhoods have fibroblasts associated with other cells, and some of them have macrophages, some of them have endothelial cells associated with them. Now I can ask, what are the differential expression patterns across these fibroblasts, right, for example, in the different neighborhoods? So particularly in this neighborhood where there's a lot of injured cells, right, how does this expression pattern differ compared to other fibroblasts? And we see that indeed there's quite a number of genes upregulated in the fibroblasts near the injured cells, particularly this gene, the CRLF1, which is a cytokine receptor. And you can see that not only does the fibroblast express this gene, but that the corresponding ligand, the CLCF1, the cytokine, is expressed strongly in the injured population. This is actually very clear from the data itself. If you look at just the spatial data, you can see the injured cells are secreting the cytokine, and then the fibroblast is making this co-receptor. So I should clarify, the CRF1 is actually a secreted receptor. It essentially acts as a cofactor for the cytokine to be bound on the CNTF, the final receptors. So you can actually validate this by antibodies. So the VCAM1 marks the injured cells, and you can see the cytokines expressed in the same cells as the injured cells, and then surrounding them, you have this expression of this co-receptor, the CRLF1 fibroblasts, and then you have expression overall of lots of these CNTF receptors around in the tissue. So, okay, so that's all great, right? But it turns out that if you treat the data as if it was just dissociated single-cell RNA-seq data, and you cluster the fibroblasts, you see that the CRLF1 fibroblasts, right, actually spans across multiple of these fibroblast clusters, clustered based on the gene expression itself. Now, but now if you take the spatial information into account, then you can see that the CRF1 falls into that spatial neighborhood very nicely. So what this tells us, right, is that treating the data as if it was dissociated actually loses information and loses crucial information about this very distinct cell population. And we make a common assumption, right, in the single cell field that these clusters give you definitive ground truth about cell types. And this is actually not true. Many times I will ask this question to the audience, right? Like if you did these cluster analysis, how many of you actually change your parameters until you guess the clusters that make sense? No one's raising their hand? I mean, everybody should be raising their hand, right? Like, when we do this analysis, we always play like with the number of genes, and the complexity and all these parameters. And if we use those parameters to match our expectations of known biology, right? How do we fundamentally make new discovery? How do we know that the new cell types of discovery is right? So we need something that's a little bit more robust. So what I propose, based on this experiment, is that we should just do a very rough clustering of about 20 or 30 major cell types, right? And then we can use the neighborhood information from the spatial data as a way to look at function, right, in these different cells, and then perform differential gene expression analysis to actually look at the molecular mechanisms and pathways. And I think, rather than thinking about spatial methods as a fancy validation tool for single-cell RNA-seq, I would argue that conceptually, fundamentally, this pipeline of doing single-cell RNA-seq clusters and then picking genes to do spatial to validate it is fundamentally flawed. And we really should think about like how to use the richness inherent in the spatial data to extract new insights. And just to extend on this point, we can go beyond just this injured neighborhoods. We can look at several other spatial neighborhoods of fibroblasts and look at their gene expression patterns. So I'll go through this quickly. You can see actually in each one of these neighborhoods, right, these are fibroblasts in different contexts of other cells. And we match it up against single-cell RNA-seq data that's collected at different time points after injury. You can see that these different neighborhoods actually correspond to different timing of fibroblast states, after injury. And that's something that actually you don't extract out from the RNA-seq data itself. But fundamentally, what you see is that within the single snapshot at about a month after injury, it actually contains multiple stages of injury, and they correspond to different signals and different recruitment of fibroblasts, injured cells, and macrophages in the tissue. Okay, so I think the take-home message for this part is that we really need to have functional measurements beyond just RNA data. Spatial is a way to get us that fundamental function information. But other modalities are very important, looking at epigenetics, looking at protein states, and so on. So the second story I want to tell you is looking at nuclear structures directly in single cells in tissues. So using the same technology, the SeqFISH method, we can look at not just the messenger RNAs, the mature messenger RNAs, as well as the introns, but we can look at the chromosome structure at the same time in the same cell. So this is actually all done in the same cell here. So this is the messenger RNA for about 1,000 genes. This is intronic states profiling for about 18,000 different nascent transcripts. And then at the same time, in the same cell, we can look at about 100,000 DNA loci. Each one of these dots correspond to one of the genes we targeted, and so we know exactly what they are. And then in the same cell, we can map about 60 chromatin marks and look at their epigenetic states of those cells and correlate the DNA loci with the epigenetic marks in the same cell. So by doing that, right, we can actually generate these tracks that looks like chip profiles or tag profiles, right, for each one of these modifications, and see how they span across. This is just across chromosome 12, but then we have it for the entire genome, roughly about between 25 to 100 kilobase resolution. This is really limited by the diffraction limited imaging of the chromatin marks and not by the DNA itself. The DNA loci are detected at about 50 nanometer resolution. The chromatin marks are detected about 200 to 300 nanometer resolution. So that corresponds to about 100 kilobases or so. So what did we find from this, right? So what we observed is that actually, if you look at where the DNA loci are with respect to these chromatin marks, the nuclear bodies, you see many of the DNA loci are consistently associated with a particular chromatin mark, right? So in the green here, for example, is the heterochromatin, and then in pink is the speckles. The DNA loci are consistently associated with them are actually occurring on the surface of these nuclear bodies. So we think about these chip profiles, we think about bead along a string with these sharp peaks, but physically in the nucleus, we're seeing that they're often coated on this two-dimensional surface on these nuclear bodies. So that's where the DNAs are localized. And another thing we noticed is that across different cells, many of these DNA loci are highly consistent between different cells. So if you're a DNA loci that's marked by H3K9 trimethylation, you're always going to be associated with H3K9 trimethylation nuclear bodies in a single cell. So for example, chromosome 4, the beginning of chromosome 4 is always associated with this heterochromatic region, and the end of chromosome 4 is always associated with the speckles. Then in every cell, you see that chromosome 4 is spanning, so the beginning part is near heterochromatin and the end part is near speckles. So even though individual cells, if you look at the chromosome structure, looks very heterogeneous. It looks like snowflakes, right? Each one is different. And if you look at the nuclear bodies, they look also very different between different cells. Now, when you integrate these two modalities together, in fact, you see that they're highly consistent between cells. That chromosome 4 is always associated with, one body on one end and another body on another end, and this is the same between many cells. So the chromosome structure in single cells is not that random. It's actually highly deterministic. And there are specific constraints you can put on the chromosomes that give this fundamental structure. Now, what about between different cell types, right? So if we do this experiment in the mouse cerebellum, in the mouse brain, we can see that the active marks are highly consistent between different cell types. So if you take these chromatin tracks and look at them and compare them between Purkinje cells and interneurons or Purkinje cells versus glia, right? The speckle marks, the active marks, are highly, highly correlated between cells. Their correlation coefficients like 0.9. But then if you look at the repressive marks, they're very different between different cell types. So the major message of this is that, the active marks seems to be, the genes are near active loci, seems to be kind of poised to be transcribed, and they're sort of stationed near these speckles or active regions, whereas the repressive regions toggles very significantly between different cell types. So if you look at Purkinje interneuron versus interneuron versus glia, the heterochromatin organization is very different in the different cell types. And you see the switching between the DNA loci are associated with these repressive compartments. So the repressive genome is actually very, very interesting in their organization. And just highlighting some of the loci here, they're associated with this repressive mark here in the Purkinje cell, and then just in your neighboring glia cell, they're now associated with the lamin. So, and this we show that it contributes significantly to the radial positioning of many of the chromosomes in these cells. So in Purkinje's, these repressive domains are in the center of the cells, and but you have a bunch of interchromosome interactions in the center of the cells. And then in the glia, they're more spread out because they're being essentially diluted out into the lamin. Okay, let me wrap up by saying that hopefully I show you a glimpse that it's possible to do discovery-based spatial genomics beyond just confirmation of these pretty picture of tissues, but you can actually get biological insights by looking at transcriptomes at the level of genomes, and then also looking at chromatin structures along with antibody marks all in the same cells. So, and that's all possible because we can scale to the level of thousands of genes and not just confined to 100 genes or so. So I want to thank the people that did the work. So Mikhail Polonsky is the postdoc that did the kidney work. Linus is the person in the lab that's responsible for scaling the method up to the transcriptome level. And then Uday, of course, the master of DNA fish, and he did all these beautiful experiments looking at chromosome structure. And I really want to thank Barbara Wold, who's a professor of biology at Caltech. And I started working on this field because of a random conversation with Barbara just when I was about starting my lab. We're just having coffee together, and Barbara said, yeah, tell me something new about single molecules. And then, so I started telling her about how, you can do like super resolution microscopy. And then by the end of the conversation, it became like something sparked, right? And it's like, maybe we can use single molecule tools, imaging tools now to do genomics. And otherwise, I would have done yeast biology. So I basically switched my direction of my lab basically right before I started my lab, and embarked on this decade-long excursion into spatial biology. So thank you for your attention, and happy to answer questions.
Thank you very much.